There are two major branches of chemistry. One branch is inorganic chemistry. Inorganic chemistry deals with acids, bases, and salts. For the most part, compounds studied in inorganic chemistry are ionically bonded, and they're all relatively small molecules. Now, there are some exceptions to the ionic bond. Water and carbon dioxide are both covalently bonded, but they are inorganic molecules. Organic chemistry deals with molecules that contain both carbon and hydrogen. That is really the hallmark of an organic molecule. It has carbon and hydrogen. Notice that water has hydrogen, carbon dioxide has carbon, but water doesn't have carbon, carbon dioxide doesn't have hydrogen, so they're not organic molecules. They are inorganic molecules. Carbon and hydrogen is what makes something an organic molecule. Now, most organic molecules are relatively large. The smallest organic molecule is methane, which is 1 carbon and 4 hydrogens. After that, they're pretty good sized molecules. Organic molecules are also made up of repeating subunits called monomers that are strung together to make polymers. So organic chemistry is sometimes referred to as polymer chemistry. Because carbon and hydrogen bond to each other pretty securely, and most of the organic molecules are just chains of carbon and hydrogen that are not very interesting to other molecules when it comes to reacting with them, at some point on every organic molecule is a special structure called a functional group. And the functional group is the reactive part of the molecule. It is where there is enough difference in the molecule that another molecule will want to react with it at that point. All organic molecules are covalently bonded. Now, as we look at acids, bases, and salts, remember, these are all ionically bonded materials, and when you put ionically bonded materials in water, they dissociate. So salts dissociate into a cation and an anion. Acids dissociate into a hydrogen ion, in other words, a special cation and an anion. And bases dissociate into a cation and a special anion called a hydroxyl group. So something like NaCl would be a salt because it would dissociate into sodium and chloride. Something like HCl would be an acid because it would dissociate into hydrogen ions and chloride ions. And something like NaOH would be a base because it would dissociate into sodium ions and hydroxyl ions. Here you see the difference between an acid, hydrogen ions, and some anion, could be any kind of anion. And here's a base, the hydroxyl ion, and some cation. Acids and bases mean that we now have to talk a little bit about pH. And the definition of pH is it's the negative logarithm of the hydrogen ion concentration of a solution. That sounds tricky, doesn't it? Well, let's worry about what we have to know about it. First of all, pH is a scale that runs from 0 to 14. There are no units on this scale. This is just a number scale. Here in the middle, 7 is neutral. There is no real pH at 7. 7 is just neutral. As you move from 7 to 0, you're moving more acid. As you move from 7 to 14, you're moving more basic or more alkaline. And the further you are from 7 in either direction, the stronger you are. So something with a pH of 7 is neutral. Pure water is a 7. Now if you go to something with a pH of 8, that would be a weak base. If you go up to a pH of 13, that would be a strong base. If you had a pH of 2, that would be a strong acid. A pH of 6 would be a weak acid. So that's really all we need to know about pH. Now here's some pH values for some common kinds of things that you are familiar with. Notice that battery acid is the extreme acid and sodium hydroxide is the extreme base. However, oven cleaner comes in at a close second for strong base. Seawater is slightly alkaline. In the olden days, before we had pH paper or pH meters, alchemists used to test the pH by tasting things. Anything that is acid has a slightly sour taste to it. Think lemon juice. Battery acid burns off your taste buds before you can register taste, so you don't notice that. Beer has a little bit of a tang to it. Tomatoes are a little bit acid. So these are things that are acid. On the other hand, things that were basic would feel slippery and taste bitter. And if you've ever had a big mouthful of seawater, you notice that there's a little bit of a bitter aftertaste. It's not a strong bitter, but you just get a little aftertaste of bitter. Organic molecules, remember, are long chains of hydrogen and carbon, and the long chains of hydrogen and carbon are not very reactive, so the functional groups act as the reaction sites. Functional groups can also categorize molecules. A lot of people think organic chemistry is hard, but once you learn what the functional group is and how that functional group reacts with everything, if you see this on a molecule, it doesn't make any difference what else you know about that molecule. You know how that molecule is going to react with anything else. Now, this OH, we've already said, is the hydroxyl group. This classifies things as an alcohol. We have ethers, this double bonded O. By the way, whenever you see an R, that's simply the rest of the chain. That's the hydrocarbon chain. That's what the R stands for. The difference between aldehydes and ketones is whether the double bonded oxygen is in the middle of the chain or at the end of the chain. Carboxyls are acids, and so forth. You do not have to memorize these functional groups. You'll just see them showing up as we start to talk about what major organic molecules are. Organic molecules have those repeating subunits that we refer to in general as monomers, and when we string monomers together, we get polymers. Organic chemistry got its name because at one time it was believed that only living things, living organisms, could produce these molecules. But in the late 1800s, somebody made urea in a test tube, and that opened up organic chemistry to a whole host of synthetic organic molecules. That's why we have Dacron and nylon and styrofoam. Those are organic molecules, but they're man-made. The organic molecules we're interested in, of course, are the true organic molecules, the ones that are made and used by life forms, and there are 4 of them: lipids, carbohydrates, proteins, and nucleic acids. So let's look at lipids first. Lipids have in them the atoms carbon, hydrogen, and oxygen. And the monomers, the basic building block, is a molecule of glycerol, that's an alcohol, and 3 fatty acids. They're held together by what are called ester bonds. Now, lipids are your fats. Their function is to act as concentrated energy storage molecules. A gram of lipid is 2.5 times more concentrated in energy than a gram of carbohydrate. And carbohydrate is our primary energy molecule. So lipids are a very concentrated energy storage molecule, but they're kind of a long-term energy storage molecule. If you're storing energy in fats, it's sort of like putting your money in a CD. It goes in, it may be a while before you take it out. It can be kind of a permanent storage if you're not careful. These ester bonds, here's the glycerol molecule with these OH groups, and here are the fatty acids. And these little points are where there's carbon, and there's hydrogen off of all of the carbons. So these are hydrocarbon chains. And here is their functional group, the acid part. And this uses your basic dehydration synthesis. We take the OH group off of the alcohol, and we take a hydrogen off of the acid group of the fatty acid, and we make water. We'll actually do that 3 times because there's 3 binding sites on glycerol, and we'll make 3 molecules of water, and we string together a lipid. This single chain indicates that it's carbon with 2 hydrogens, carbon with 2 hydrogens, carbon with 2 hydrogens, because carbon is looking to make 4 bonds total to keep its electron in the outer shell the same. But occasionally a carbon will double bond with itself, and you'll have fewer hydrogens there. And when that happens, you get a bend in the chain that kind of throws the chain off. So These two fatty acids were saturated. They had all of the hydrogens on them they could. This particular fatty acid here, this last one, was an unsaturated fatty acid. Because it has the double carbon bond, it doesn't have the maximum number of hydrogens associated with it. Here are some examples of those saturated and unsaturated fatty acids. Where we have single bonds between all the carbons, we're going to have the maximum number of hydrogens. These are going to be saturated fatty acids. They have as many hydrogens on them as they can. Most saturated fatty acids are from animals, and they tend to be solid at room temperature. As we move into the unsaturated fats, if there's only one double bond in the chain of the fatty acid, then it's monounsaturated fat. If there's more, then it's a polyunsaturated fat. These tend to be from plants, and they tend to be oils at room temperature. Now, at one time, saturated fats were thought to be very, very bad. Saturated fats do drive you toward producing cholesterol a little bit more in your liver, and so margarine became very popular. And to make it solid, because in this country we don't like using oil, we like a little bit of spreadability to our fat, they would saturate the unsaturated fats. The only problem with this, we've now discovered, is the saturation, the artificial saturation of an unsaturated fat gives you a saturated fat that is even more unhealthy than regular saturated fats. So margarine has now fallen out of favor because of the trans fats that are in them. There are some specialized lipids we need to talk about. Phospholipids are glycerol and two fatty acids, but one of the fatty acids is taken off and a phosphate group is added. This means that the glycerol fatty acid end is hydrophobic. It doesn't like water. It moves away from water. But the phosphate group is hydrophilic. It will be attracted to water. This makes it settle up in a way, when you put it in water, that makes it very good for making up cell membranes. So phospholipids are a major component of cell membranes. Waxes we see in the cell walls of some organisms. And this is one long-chain fatty acid that's linked covalently to a long-chain alcohol. So it's different from your regular lipid in that there's only one alcohol and one fatty acid, but the alcohol has many more carbons in it than the glycerol. There is no hydrophilic head, so waxes are completely insoluble in water. Steroids are four carbon rings that are attached to each other, and different side groups will be attached to make different steroid type molecules. Steroids are primarily used to strengthen cell membranes. So as we look at our phospholipids, here's the glycerol, here's the two fatty acids that are attached, and then one of those fatty acids has been replaced with this phosphate group. Now this phosphate group will interact with water, but the lipid tails are what are hydrophobic. They don't like water. So when you put phospholipids in a solution, they automatically line up like this. Those little hydrophobic tails seek each other out, and the hydrophilic ends, phosphate ends, face outward. This makes it an excellent component for the cell membrane, giving you sort of a barrier between the inside and the outside. Cholesterol is that basic four-ring structure, and here is actual cholesterol. These are the extra pieces that are hanging off of it. And you can see if you were to build this out of something like Tinker Toys, this would be really hard to change shape. This would be a very stiff kind of molecule. And so we interweave cholesterol into cell membranes, animal cell membranes anyway, to give it some strength. In other cell membranes, there are other sterols other than cholesterol. Our second organic molecule are carbohydrates. Now, carbohydrates have the same atoms in them as lipids, carbon, hydrogen, and oxygen, but they're arranged differently. The monomer for carbohydrates is a monosaccharide, and monosaccharides are held to each other by glycosidic bonds. When you put two of them together, you have a disaccharide, and as you put more of them together, you come up with a polysaccharide. Carbohydrates can function as structural components. Chitin, cellulose, peptidoglycan are all structural components. If you're not sure what chitin is, if you step on a roach in the middle of the night and it goes crunch, chitin is what makes it go crunch. It's that part of the exoskeleton that makes the exoskeleton hard. Cellulose is what is in plant cell walls. It's what makes celery crunchy. And we've mentioned how peptidoglycan is a component of the cell wall of bacteria. It's also the primary nutrient and energy source for organisms. The two primary carbohydrate molecules that are energy sources are glycogen and starch. You can tell you're dealing with a carbohydrate because it ends in ose. That's your monosaccharide. It'll be some sort of sugar ending in OSE. Now, we typically use the number of carbons in the monosaccharide. So if there's 5 carbons, we call it a pentose. If there's 6 carbons, we call it a hexose, rather than speaking about a specific sugar. 2 monosaccharides make a disaccharide. Multiple monosaccharides make a polysaccharide. So here you see, again, we use dehydration synthesis. 2 monosaccharides. These are 2 hexoses because there are 6 carbons in them. We can take an OH away from one and a hydrogen away from the other. We have to put in some energy, of course, because we're making bonds. Here's what we mean by a glycosidic bond. Here's how they're linked together. And we get water as a byproduct. This is a dehydration synthesis. This is making sucrose, which is glucose and fructose. If we reverse that, we want to break this bond, we have to have water, because then we make back the glucose and the fructose by using the hydroxyl from the water and the hydrogen from the water to turn them back into their individual monosaccharides. Cellulose is this kind of framework of glucose molecules. Starch is a long unbranched chain, and glycogen is a long chain. So all of them use glucose molecules. They're just held together differently, and that's what gives them their differences. You can see how this grid of glucose molecules would be a very long structure, but how these chains, it would be easy to take off a glucose and use it for energy, and take off a glucose and use it for energy. The third of the organic molecules is the protein. Proteins contain carbon, hydrogen, oxygen, and nitrogen. Some of them contain a little sulfur and some contain a little phosphate. The monomer is the amino acid. There are 20 different amino acids, and some of the amino acids are the ones that contain sulfur and phosphate. Amino acids all have this basic structure. They have the amine group, this nitrogen and 2 hydrogens, and then they have this carboxyl group, this acid group. And these are attached to the same carbon. Then what makes each amino acid different is whatever this R group is. So here is phenylalanine that has a ring structure. Here is cysteine. It just has a sulfur hanging out there. Glutamine has a little short chain of carbon with another nitrogen out there. Serine has a hydroxyl group on it out there. But this is what makes up the backbone of a protein, these amine and acid. So literally, this is an amino acid, is what links together to make up your proteins. Proteins are held together with peptide bonds. You make dipeptides when you string 2 together, and then you make polypeptides. So that again, using dehydration synthesis, we take the OH off of the acid group, an H off of the amine group, and stick them together. We make water, and now we have a dipeptide. Because this particular amino acid only had, here's its remaining group, whatever it was. And the way this goes together, the other remaining group is stuck on this side of the molecule. Now this is important as we talk about protein structure. Proteins also have 2 functions. Proteins always go together and have a very, very specific shape to them. As a result of this shape, this is what makes them perform the function they perform. Sometimes the shape is structural, so that it's just a nice, solid brick kind of molecule that fits in and helps make flagella or the cytoskeleton or some of those kinds of things in a cell. But a number of proteins are functional. We've mentioned that all chemical reactions in your body have to be controlled by a catalyst, and in living cells, that catalyst is a protein called an enzyme. So proteins are key to all of the chemical reactions occurring in any cell. Proteins also, many of your hormones are proteins, so they can be involved in regulating cell activities. They act to transport molecules through the cell membrane, and they act as defense. Antibodies and complement are special chemicals found in the blood that help protect against disease. Now, protein structure is key to how a protein functions, and there are 4 levels of protein structure. Primary structure is simply the exact amino acid sequence, and this is critical. There are some instances where changing one amino acid in a chain of 800 amino acids makes a critical difference in how that protein functions. For example, the difference between sickle cell hemoglobin and normal hemoglobin is the substitution of one amino acid different in the 6th position on the beta chain of the hemoglobin. That one little changes from a functional molecule to a less functional molecule. Now, the secondary structure has to do with those R groups that were sticking off on either side of that peptide bonding string. Those R groups, whatever the rest of the molecule is that's sticking out there, can start producing ionic bonds or hydrogen bonds in between them. So you've got the basic protein backbone of peptide bonds, and then you've got these other parts of the molecule that can kind of twist up a little bit and bond with each other. You get either a beta pleated sheet or an alpha helix. Those are the 2 structures that you have in the secondary structure. As a result of this bending of the secondary structure, it brings some R groups from amino acids close to other R groups that were maybe 20 or 30 amino acids apart originally. And so this starts a very complex folding piece, and here there are covalent bonds and disulfide bonds that are starting to form between the rest of any of the amino acid molecules. Now, quaternary structure occurs if 2 or more polypeptide chains get held together by covalent bonds or by other molecules. An excellent example of quaternary structure is hemoglobin. Hemoglobin is 4 polypeptide chains that are all attached to an iron molecule, so that gives you a quaternary structure. So our primary structure, just that basic peptide bonding chain. But you see how the rest of the molecule is hanging off here and here and here. And so they can start coming in contact with each other here, or they may twist around here, and you start getting this secondary bonding. And this either coils it up into the alpha helix, or you start getting this folding back and forth in what's called the beta pleated sheet. But once either of these happen, now different R molecules are close to each other, and they can start to bond with each other. So you get these pleated sheets and these alpha helices, because you can have both of them in one long string of amino acids, and they start really folding up, and it gets very, very convoluted. If you have 2 or more of these polypeptide chains linking up with each other somehow, either covalently bonding to each other or joining with another molecule that holds them together, then you have your quaternary structure. Proteins rely on their particular shape, but if you damage their shape, you do something called protein denaturation. You denature the protein, and that's altering that secondary or tertiary structure. You have to break the bonds between the amino acids to alter the primary structure, but the secondary and tertiary structure is much easier to disrupt. It can be disrupted by a change in pH, by mechanical agitation of the protein molecule, by heat, or by ionic imbalance. Now, if you would like to just experiment with protein denaturation a little bit, take an egg white. Egg whites are composed of albumin, which is a protein. They're almost pure protein. You can put a little bit of egg white in some vinegar. Vinegar is acid, so that's a change in pH. Observe it for a little while. You'll see it get kind of white and coagulated looking. You can take an egg white and you can beat it, and it'll get all foamy, and if you beat it long enough, it'll get foamy and stay foamy. Now, if you really want to stabilize that foaminess, throw a little cream of tartar in there. Cream of tartar gives you ionic imbalance, and that will even stabilize further the disruption of the protein by the mechanical agitation. And then I'm pretty sure you've all seen egg white after it's been cooked. It looks a lot like you're going to see with the egg white that goes in the vinegar. So those are all examples of protein denaturation. Once you have done that, these are forms of irreversible denaturation. That albumin is never like it was before. It's changed, it's altered, so it has a different structure to it now. So when you denature protein, you alter its structure and you're going to alter its function. Nucleic acids are the fourth group. Here we have the atoms carbon, hydrogen, oxygen, nitrogen, and phosphate. The basic building blocks, or the monomers, are the nucleotides. Nucleic acids are molecules that function in managing information in the cell. A basic nucleotide is made up of a phosphate, a pentose, a 5-carbon sugar, and you've got your choice of deoxyribose or ribose, and a nitrogen-containing base. And you've got 5 choices here: adenine, guanine, cytosine, thymine, or uracil. So as we look at your pentose sugar and your phosphate, and then here are your bases, and they're divided into what are called the purines and the pyrimidines. The pyrimidines are a single ring, the purines are a double ring. So there's your sugar. Here are your bases. You have a phosphate group stuck to a sugar, and then you stick one of these bases here, and you have your nucleotide. There are 2 nucleic acids. Deoxyribonucleic acid, or DNA, has as its pentose, surprise, surprise, deoxyribose. It tells you in the name what its pentose is. They all have phosphate, and there are one of 4 bases in DNA: adenine, guanine, cytosine, and thymine. Now, we string nucleotides together to make a string of nucleotides, but DNA is a double helix. It's a double string of nucleotides, and the nucleotides are held together between the bases by hydrogen bonds. DNA, as you know, stores your genetic information. So if we look at a DNA molecule, here's your basic string. The phosphates and the sugars join up to make the backbone, and then the bases are strung off here. And adenine will double hydrogen bond to thymine, guanine will triple hydrogen bond to cytosine, and those hydrogen bonds will hold these 2 strings of nucleic acids together, and then it coils up like a spiral staircase. That's the double helix part. Remember, hydrogen bonds are simply weak attractive forces, so it's pretty easy to break these bonds, which is convenient because we spend a lot of time zipping and unzipping DNA to use it to get to the information we need to. Now, the other nucleic acid is ribonucleic acid. The pentose is ribose. There's the phosphate. Again, you have your choice of 4 bases. Adenine, guanine, and cytosine are the same, but instead of thymine, RNA uses uracil. RNA also differs from DNA in that it's a single chain, and it differs in its function. RNA is involved in translating the DNA into protein, and we'll find out later that there are 3 kinds of RNA, and we'll talk about how the DNA and the RNA react to make the proteins. Now, your RNA molecule, single strand, so those bases are just sticking out here to the side. They don't pair with anything. They're just available so that we can use them to transfer information. Adenosine triphosphate, or ATP, is a very special nucleotide. It stores energy in chemical bonds. Now, we talked about lipids being energy storage molecules, but they're more like a CD or a savings account. It's harder to get your energy out of those kinds of situations. ATP is a little bit more like your checking account or your debit card. You put money on it, you take money off of it. It's very quick. You don't have to go through a lot to get your money back. The same is true with the energy and ATP bonding. Now, adenosine triphosphate starts out as a basic RNA nucleotide. It has ribose, adenosine, and one phosphate. Phosphates have a little bit of a negative charge on them. So if we try to stick a second phosphate on and make adenosine diphosphate or ADP, it takes a little extra energy to get that bond to make. So we have some extra energy stored in that bond. And by the time we try to put a third phosphate on, because now we've got two negative phosphates already on the molecule, and we're trying to stick a third negative phosphate on there, it really takes a lot of energy. So there's a tremendous amount of energy in that bond. So if you look at your basic nucleotide here, adenine and the ribose and the phosphate, your AMP, that's your basic RNA nucleotide. When you stick that second ADP on there, notice the wavy line. That indicates that it took a little more energy because we're sticking a negative to a negative. And by the time we put that third phosphate on there, we're sticking a negative to two negatives. So another wavy line indicating that we have a tremendous amount of energy in that bond. When we remove those phosphates, we release that energy. So this is a high energy storage bond. ADP plus energy plus phosphate gives you ATP, and ATP breaks down to a phosphate and that energy and ADP. And usually this is the flow. However, ADP can also be broken down to AMP, releasing some more energy from that bond.